In my previous session with you, I discussed general aspect of biotechnology and its historical perspective. And what we understood that biotechnology deals with uh, developing technologies using biological principles to develop products and services for the benefit of humankind. So therefore, it's very important to understand what is this biological principles and how they have been utilized for developing technologies. So, in other words, one has to understand the biological system existing in an organism. And when I say that, what actually I mean is defining functions in an organism biochemically and also finding out their genetic basis. So if we can link the biochemistry of a function to the genetic basis very clearly, then we can think of manipulating this function at the genetic level in order to manipulate or alter that function. You know. And to achieve all this, it is very important that we understand what is the basis of a living organism. So today, what I'm going to do is to take you through very quickly to this basis of living organism. Okay, so as you know, there is variety of living forms exist, which you can see, some of which you can see, and others you cannot. In the sense that you have living forms which are visible to you, which you can see around. You have animals, you have plants, and then you have large number of living forms which you cannot see. But all of them are made up of with a unit what we call cell. Okay, so cell is the basis of life of all living organisms. Okay, now whether it is a unicellular organism or a multicellular, multi-organ organism, cell remains the basis of life or unit of life. Okay, now based on the knowledge generated previously, SNAP, a cell theory was proposed. You know, and what is this cell theory? So the cell theory has three important aspects. One is that all the known living forms, they are made up of one or more than one cell. Okay. And secondly, very important, that all living cells, they arise from a pre-existing cell by division. So life can only be created by a living cell, not by any other means. Okay. And the cell is the unit of structure and function in all living organisms. So this was the cell theory proposed, that all living forms are made up of cells, and the living cells, they arise from a pre-existing cell by cell division, and the cell remains the unit of structure and function in an organism. Okay. This is very interesting, you know. And if we look at the historical part of the cell theory, as I give you little bit hints in my previous lecture also, so it's not new, the concept, I mean, how the concept was built up, you know. So dates back to 1665, where Robert Hooke discovered the cell, okay? And subsequently, it is the Leeuwenhoek who observed a living cell, you know, and much later, it was Robert Brown who discovered a nucleus inside a cell, you know. So we are talking about uh, a journey of 200 years just in discovering the cell, observing a living cell, and and nucleus being identified, you know. Subsequently, if you see, uh, within the cell, a fluid-like structure was discovered by Dujardin in 1835. Okay, and very interestingly, Sheldon was the first one to look at plants and proposed that all plants are made up of cells, you know. And subsequently, in 1839, it was Perkinje who gave the name that fluid, which was present in the cell, as protoplasm. Okay, so we have a progression in our knowledge, you know, about looking at living cell, observing nucleus inside it. Then plants are made up of cells. The cells have protoplasm inside, and subsequently, Schwann said that animals are also made up of cells, you know, and in about 6 years later, you know, Brown proposed that the cell is the basic unit of life, you know, and it was Virchow who said that cells arise from a pre-existing cell, you know, cells arise from a pre-existing cell. So that is the timeline, you know. So in 1855, the cell theory was put together, which I gave you in the beginning, that uh, living beings are made up of cell, and the cell arises from a pre-existing cell, and cell is the unit for structure function of all organisms. So, starting from 1600 to 1855, you know, and almost around 200 years, this knowledge was put together in the form of cell theory. So, if you look at, as I told you, we have unicellular organisms, we have multicellular, multi-organ organisms, which are divided into prokaryotes and eukaryotes. So these are some of the examples. If you see, you know, Euglena, for example, Paramecium, yeast, they are lower animals and the lower plants, which are unicellular, and they perform all the functions of the living organisms. They multiply, they reproduce, you know, they move about, you know. So these are the examples of the unicellular organisms. And then you have multicellular organisms, you know, for example, plants, like animals, a whole range of animals. And then you have fungus, which sits in between animals and plants, but classified as plants, you know. So these are some of the examples which you see of living forms, unicellular to multicellular organisms. So therefore, uh, when the people started looking at these cells, very interesting facts came into uh, existence. For example, if you look at the size of a cell, it varies. You know, again, a single cell you cannot see with naked eye. But then, if you see here some of the examples, for example, if you look at uh, the smallest cell is the mycoplasma, which is about zero point 0.1 micron. And then you have the sea urchin egg, which is so big, and it is a single cell, so 18 centimeters. So you have so much of variation in the size of a cell, from microscopic to macroscopic, you know, and then the shape varies. Uh, Again, the example from human system, if you see here, the smallest cell is the sperm cell in humans, which is about 5 micron, to the largest cell, like the ovum, which is 120 micron or micrometer. And the largest cell in humans is the nerve cell, which is 1 meter. So from very small to big, and also the, the size varies, as I mentioned, within the organism and within different organisms. And also, if you look at the shape also is different. If you compare RBC nerve cells and the WBC, they look so different. So, so the point I'm trying to make here is that the basis of life or the unit of life differs in size from microscopic to macroscopic in their shapes. They're different depending on the kind of function they perform. Now, how this knowledge was generated is based on these two tools, simple microscope to compound microscope. This is the compound microscope, and the electron microscope. This is the electron microscope, and this is the compound microscope. How they differ? They differ in the power of magnification. A compound microscope can magnify up to 2000 times, and an electron microscope can magnify 500,000 times an object. So basically, it is after the discovery of electron microscope, we gained knowledge about the cell structure, cell organization, internal organization of the cell was very clearly defined and refined only after discovery of electron microscope. Okay, so people were able to look inside the cell, what is present within the cell. And in next couple of slides, we will quickly go through, how uh, three different types of cells we now know how they look like. For example, animal cell, plant cell, and a bacterial cell. Okay, so all the three types of cells have been studied structurally and functionally in general. So you are familiar with this slide. I mean, I will just go through. This is the typical animal cell, if you look at. So it has a nucleus, and then you have Golgi complex, that is Golgi complex, endoplasmic reticulum, and the mitochondria, you have lysosomes, you have cytoplasm, and here endoplasmic reticulum, you have rough and smooth surface, and So this is the typical structure of a animal cell, which is surrounded by membrane, plasma membrane, what we call, it is encapsulated, and within that you have these many organelles, and all these organelles perform any specific function within the cell. Okay. Now, if you look at a typical plant cell, it also has some common organelles and some very specific to plant cells. For example, here you have cell wall. The first covering is the cell wall, and then the cell membrane, or plasma membrane, which is common to both animal and plant cells. And you have nucleus, you have mitochondria, you have endoplasmic reticulum, similar to animal cell. And what it contains is this green structure, what we call chloroplast. Now, the chloroplast is missing from animal cell. And also they have vacuoles, large size vacuoles in plant cells. So this is the typical plant cell structure, or that's how plant cell organizes itself. And coming to bacterial cell, again, it has an outer covering, which is cell wall, and then cell membrane, But what it lacks is the cellular organelles, well-defined membranous organelles. There is nothing which we saw in animal and plant cells typically. It has chromosome, which is single, they don't have nucleus, but a nucleoid or nuclear-like structure wherein they have the genetic material, and all other necessary functions are carried out loosely within the cytoplasm. And they have flagella, pili, and all these structures for movement outside the cell. Okay. But majorly, the bacterial cell, as I mentioned, doesn't have a well-defined membranous organellar structures. Okay. They have cell membrane and then the cytoplasm. And in that, the nucleoid structure floats around, and they have food granules and plasmid DNA. Plasmid DNA and the nuclear DNA. Okay, now if we look at each of these, or if we, try to look at what typically a cell has or consists of. So you have plasma membrane, which is common to all cells. You have nucleus, which is characteristic of a eukaryotic cell. Then you have cytoplasm, and in cytoplasm, you have these cellular organelles like endoplasmic reticulum, Golgi complex, lysosomes, vacuoles, mitochondria, plastids, centrosome, and cytoskeleton. These are the organelles, and this is the typical, eukaryotic cell organization, and how these organelles organize themselves structurally, that is also very clearly understood. If you look at the plasma membrane, is the bilayer structure, which is made up of proteins and the lipids. These are the two major biochemical molecules which makes a cell membrane, and and the remaining cellular organelles are manifestation of this plasma membrane, which is wherever it is a bilayer membranous structure in organelles as well. Okay, and this forms the outermost covering and encapsulate all the organelles together, gives shape to the cell, and helps in communicating. Cell-to-cell communications are there, you know. And then in plants, the outermost covering is the cell wall, as I mentioned, and that's the difference between animal and plant cell. The presence and absence of cell wall is the major difference. We will talk about that later. But then this covering or envelope is basically made up of carbohydrates, the complex carbohydrates, which is pectin, cellulose, hemicellulose. You know, this is the structure and this is the plasma membrane. So they organize themselves. And what is the function of cell wall in plant cell is basically it gives rigidity to the cell. Now, rigidity is important, you know. Plants do not have a skeletal system which gives shape in animals. The skeleton system gives shape to an animal. Plants don't have. But the cell wall and billions of cells, when they unite together uh, through cell wall, it gives rigidity to the structure, you know, and and also helps in giving shape and strength, as I mentioned, and it prevents cells to dry out, you know, and also protects cells from external pathogens also. You know? So very important organelle as far as plant cell is concerned. If you look at the nucleus within the cell, and if we go deeper inside to the nucleus, you have nuclear envelope which covers it, and inside the nucleus you have nucleoplasm, that is the fluid-like structure, and you have chromatin and then nucleolus, you know. And interestingly, the nuclear envelope is not a continuous structure. It has a lot of pores in it, so nuclear pores, and that is because of the movement of the macromolecules from nucleus to cytoplasm and cytoplasm back to the nucleoplasm. Okay, so this is the broad organization or typical organization of nucleus in eukaryotic cells, whether it is an animal or plant. This is the organization of it, of the nucleus. And what it contains inside, you have these thread-like structures, what we call chromosome. And chromosome is the storage house of genetic information, and how that is in turn, the chromosomes have these fibers like chromatin, and chromatin has, you know, what is made up of protein and DNA. Okay, and on DNA, the genes are located. So that's how the genetic information is condensed into the structure and takes a shape like this, and we call them as chromosome. Okay, and this is very important, and we'll be talking about it later, because it is here that the information about cell function, what a cell is going to do singularly or collectively together in an organism, is guided by the information residing in nucleus on chromosomes, and in turn on DNA, and in turn the genes which are coded by the base sequences on DNA. You know? So that's how it's very, very important organelle of the cell in all cell types, whether it is plant or animal or bacterial cells. Okay. So cytoplasm basically is the fluid which is encapsulated by cell membrane, and in which all these organelles float around, and it's a semi-viscous solution, jelly-like material, and then you have the endoplasmic reticulum. So it's a network. Again, all these are membranous structure, as I mentioned, double membrane, and it fills the space within the nucleus, and they are the site of protein synthesis. You have rough ears and the smooth ears, and it's there on rough that the proteins are synthesized, and are taken to their site of action, wherever they are supposed to function. The proteins are synthesized here and are then translocated to their site of action. And you have again another membranous structure, what is called Golgi body or Golgi complex. This deals with the cleaning up of the cell, where whatever waste material is generated, it is thrown out of the cells through this organelle, what we call Golgi complexes. So you have incoming and outgoing trafficking is regulated by Golgi complexes. And then you have lysosomes. These are small membranous structure, just single membrane covers, and then here the lot of toxins which are created by cellular functions are taken in here and degraded and thrown out of the cell. So basically, it is it is for the cleaning purposes that this organelle exists. And you have vacuoles, as I mentioned. Vacuoles, plant cells have bigger vacuoles as compared to animal cells, and they are the storage houses. Again, whatever cell doesn't require is put into the vacuoles, and whenever they require, they take it back. So in plant cells, they have a huge role to play. Again, this is a membranous structure. It's a membranous structure. You know, and then, okay, so in addition to this, these organelles, there are two major organelles in the eukaryotic cell, and one is mitochondria, and another is plastid. Mitochondria basically is the site of power generation. Okay, here the complex carbohydrates are degraded, and while degrading, they release energy in the form of ATPs, which is utilized for other functions. So it's a typical structure, again, membranous structure, and all these things we have studied. I'm not going into the details of the functions. In the details, you can revise from your previous notes. But then, if we come to plant cell, which has a very specific organelle, what we call plastids. Okay, plastids are basically of three types. You have chromoplast, chloroplast, and leucoplasts. Chromoplasts are the colored plastids. Chloroplasts green in color, very specifically, due to the presence of chlorophyll, and leucoplasts are basically colorless, And you can see the manifestation of the presence of these different plastids in different parts of the plant differently. For example, you see, for example, carrot, uh, they are orange in color. Okay, that is because of the carotene, the chromoplast, and the presence of carotene gives this color. In mangoes, the presence of xanthophore and the chromoplast gives you yellow color. Okay, and tomato is lycopene. That's the pigment which is present in chromoplast and gives the red color. So that's the beauty of different kinds of plastid, like in chromoplasts, they give different colors depending on the kind of pigment they contain. And then you have leucoplasts. These are the examples. That is because of the presence of the stored material. For example, in potato, it's the starch storage, so it gives this kind of color. Maize grains, proteins generally, this color, and the oil storage seeds, they give this kind of color, Glucoplast. Okay, so in plants, we have plastids, which are divided into three groups: chromoplast, chloroplast, and leucoplast. Okay, and finally, if you look at the chloroplast, so chloroplast is green in color, and the greenery which you see around in plants is because of this presence of chloroplast in the leaf. Okay, so a typical structure of chloroplast is like this: the double membrane structure, inner membrane, outer membrane, and then stroma, thylakoid, and basically it is the the synthetic organism in the sense that it is responsible for synthesizing uh, carbohydrates from simpler forms, water and oxygen, water and carbon dioxide, converted into carbohydrates from simple molecules to complex molecules with the help of chlorophyll, or harvesting the solar energy through chlorophyll by a process we call photosynthesis. So that is the side, you know. And in addition, let me tell you, both mitochondria and chloroplasts have their own genome, what we call blastome, and that is the chloroplast genome, and mitochondrial genome. Uh, but then both these genomes are not exclusive. They are semi-autonomous in the sense that they do not have the complete set of genetic material which controls the functions in mitochondria or in chloroplast. So they depend on nuclear genome contribution for their function, and therefore they are referred as semi-autonomous organelles. They are not completely autonomous organelles as far as their genetic material is concerned. And we will look at what this genome consists of and whether they code for any specific trait in addition to the nuclear genome, which we will be studying in greater detail later. Okay. And in addition, the animal cells have this cytoskeleton, which is which gives strength to the animal structure. That is because of the presence of the cytoskeleton in the cell. Okay. So, to summarize this discussion on organization of the unit of life, that is the cell. So, if we look at what is the difference between prokaryotic cell, that is the bacterial cell, and eukaryotic cell, you know, so it's very simple in the sense that in prokaryotes, nucleus is underdeveloped, and in eukaryotes, nucleus is well developed. Okay, so the first difference is at the nucleus level. Second is in prokaryotes, only single chromosome, single circular chromosome at DNA, and in eukaryotes, it's more, one or more than one chromosomes are present. And in prokaryotes, the membranous organelle structures are missing, whereas in eukaryotes, there is membranous, it is membranous organelles are there. Size varies, microbial cell is very small, whereas the eukaryotic cell has, it is larger in size. Okay. As I mentioned, examples are bacteria and organelle for prokaryotes, and eukaryotic cell is all over, from plants to animals. Okay. So these are the sort of differences between prokaryotic and eukaryotic cells. And among eukaryotes, you have animal cell and the plant cell. So how these two cells differ, you know, uh, basically, it's the cell wall. you know, which is present in animal cell and absent in uh, animal cell. And plant cells have chloroplast or presence of plastids, whereas in animals, plastids are absent. So if you have to give one or two differences in the animal and plant cells, then cell wall and plastids. In addition, the minor differences, size, smaller, bigger, vacuoles, smaller, bigger, centrioles present and absent, of course. So centrioles, plastids, and cell wall, you know. So that is the major differences. Centrioles are present in animal cell, absent in plant cells. And cell wall and plastids are present in plant cells, absent in animal cells. Keep this in mind. These are the three major differences between animal and plant cells. Okay, so to conclude, what I'm trying to do is to just revise with you. I'm sure you have gone through these cell organization structure a couple of times in past semesters, but then just to build up from here, uh, we will now look at the nuclear organization and first, and then the organization of the organelle genome like chloroplast and mitochondrial genome, and understand how these genomes organize themselves, particularly the nuclear genome, and then how it expresses itself in a cell, and and then how this expression is then associated with the functions within the organism, and how the genetic information contained in DNA is transmitted from one generation to another, and within the same generation in the cell, from genes to proteins and proteins to functions. So with this, I conclude this part. Thank you very much for viewing this video.